Hello class, this is Delay Olson. I'm here to talk about the post-absorptive state with you. So this last pre-lecture video is going to really complement what we talked about in class for the absorptive state, and we'll kind of tie in some things with the absorptive state, and we'll kind of show you how they work in the post-absorptive state. I will go over these slides again in class to kind of solidify the information, because I think this is kind of an interesting and very, very physiological application of what we talked about. So let's get started. So just to review and recap what I talked about in the last lecture. In the last lecture, I talked about it was the fed state, right? So up here, look at the fed state. Here, let me get my laser pointer out. Here's the fed state here. Adipose tissue and skeletal muscle in the fed state respond to insulin. I told you that. Now, when they respond to insulin, that means glucose levels are high in the blood. So if glucose levels are high, you get insulin released from the pancreas. Insulin circulates in the bloodstream, binds to receptors present on adipose tissue or skeletal muscle. When it binds to either one of those tissues, it activates a signaling pathway that culminates on the insertion of glucose transporters into the membrane. These glucose transporters, when they're in the membrane, facilitate the movement of glucose from high concentration in the blood down a concentration gradient into the cell, and glucose enters the cell. Now, this is really important because glucose, when it enters the cell, lowers plasma glucose levels, remember, which is a toxin in the blood, and so at high levels. So what you want to do is you want to activate skeletal muscle and adipose tissue to cause them to pull in lots of glucose from the bloodstream. This helps lower blood glucose levels back down to the normal state, and you have your kind of classic negative feedback. So the take-home message for this is that in adipose tissue, fat tissue, and muscle, glucose transporters are inserted into the plasma membrane in response to insulin binding at the surface of the plasma membrane. All right, now let's contrast this to when you are fasting. Now, in the fasted state, that's what we're talking about today. In the fasted state, let's think about what's happening with adipose tissue and skeletal muscle. Now, in the fasted state, glucose levels should be normal in the blood. You always want to have some glucose in the blood, so there should still be glucose there. But is insulin present? I hope you recognize that if glucose levels are normal, insulin should not be secreted. High glucose levels will stimulate insulin secretion, but normal glucose levels should not stimulate insulin, so insulin levels should be low. If insulin is not present in the blood, that means that it should not be binding to a receptor present on adipose tissue and skeletal muscle, and that means that these GLUT proteins, these glucose transporter proteins, these facilitated diffusion transporters, should be inside in these vesicles, not at the plasma membrane. All right, so do you see how now this tissue, adipose tissue and skeletal muscle, does not have access to the glucose present in the bloodstream because it doesn't have any glucose transporters. If there are no glucose transporters, that means you can't move glucose into the cell because glucose will not cross the plasma membrane unless there is a transporter present. Okay. So the take-home point for this is that in the absence of insulin, glucose transporters are removed from the plasma membrane and sit in vesicles waiting for the next insulin stimulation. Essentially, waiting for the next meal where glucose levels will rise, insulin is released, insulin binds, glucose transporters move to the membrane and move that high level of glucose back into the blood, out of the blood and into the cell. Now, therefore, adipose tissue, which is fat, and muscle do not have access to the glucose unless insulin is around. That is important because remember what we are doing in the fasted state, and maybe I shouldn't say remember, you haven't had this yet, but in the fasted state, the glucose that's present in the bloodstreams, we are saving for the brain. And so your brain always has to have a certain supply of glucose. That's why there's always glucose that's present in our bloodstream, because the brain, there are certain parts that absolutely have to have it. And so when you're in a state when you're not eating, and especially when you're not eating glucose, what you want to do is you want to preserve the glucose that's there for the brain, and you want to limit the other tissue's ability to grab that glucose and oxidize it. And so the absence of insulin is one way that prevents both adipose tissue and muscle from accessing the glucose that's present in the bloodstream. Super cool. Now, in the fed state, we didn't really talk about this, but this is where you're going to get this information about the liver. So the liver is also insulin sensitive, meaning that it will respond to insulin, or meaning that it has insulin receptors present on its surface. Now, the reason why I didn't talk about the liver is because it works in a slightly different way. So insulin is around, obviously, when glucose levels are high in the plasma. So insulin can bind to the insulin receptor on liver, but it will do a slightly different thing here. So when it binds to the insulin receptor on liver, it will activate a pathway that converges on the activation of the very specific protein called hexokinase. Hexokinase is the protein. It's a kinase. Look, if kinases phosphorylate things, hexokinase phosphorylates glucose upon entry into the cell. And that phosphorylation event is very important. Okay, so what does it do? Let's just take a step back and take a look here. First of all, I want you to recognize that glucose transporters are present on the membrane all the time in liver. Okay, there's no movement of glucose transporters to the membrane. They're already there. So if glucose levels are high on the outside in the blood, then glucose is going to diffuse down into the cell, down a concentration gradient, and into the liver cell. Now, once it's inside of the cell, glucose levels will begin to rise. And if glucose levels begin to rise, then do you see how that might limit the movement of more glucose into the cell? In order to kind of overcome that, what the liver cell does is it phosphorylates the glucose upon entry. And when you phosphorylate glucose, it kind of tricks the system into thinking glucose levels inside of the cell are low, because glucose is not the same as phosphorylated glucose or glucose 6-phosphate. That's the actual phosphorylation site. So glucose becomes glucose 6-phosphate because hexokinase phosphorylates it. And so what happens is you increase the amount of glucose 6-phosphate in the cell, and that will go through glycolysis and eventually will either be oxidized or converted to triglyceride. But the idea here is that you maintain a concentration gradient such that more glucose can move into the cell. And insulin in this case isn't activating glucose transporters. Instead, it activates hexokinase, and it activates this enzyme that can phosphorylate glucose. And if you activate more of that enzyme, you get more phosphorylation, and you keep glucose levels low inside of the liver cell, and you maintain the inward movement of glucose into the cell. So the take-home message is that in the liver, glucose transporters are always present in the plasma membrane. And insulin stimulates the activation of hexokinase. Okay, this is different than muscle and adipose tissue. It stimulates hexokinase, which is an enzyme responsible for phosphorylating glucose upon entry into the cell. And that phosphorylation event changes glucose such that the glucose on the outside, the concentrations are high, and keeps the glucose levels on the inside low so that you can maintain an inward movement of glucose from the bloodstream into the cell, removing excess glucose from the bloodstream in the fed state, which is what we're in. You've just eaten a lot of glucose. Okay, and remember, high levels of glucose in the bloodstream are toxic. So this is one way where the liver can kind of remove glucose out of the bloodstream, preventing extended periods of elevated glucose levels in the plasma. Now let's compare that to the fed state in the fasted state. So now you're not eating. Now, and this is what you don't know yet, is that the liver is responsible for producing glucose in the fasted state. So if you haven't eaten glucose for long periods of time, remember, there's one organ in your body that absolutely needs to have glucose in order to survive, and that organ is the brain. Glucose, you have to maintain a certain steady state of glucose because the brain always has to be able to dip into the bloodstream and grab glucose. So in the fasted state, when you might not be eating glucose or when you're not eating anything, glucose levels might tend to fall. And so what happens is there's this switch. Insulin disappears because glucose levels aren't elevated anymore. So now the liver, so now the skeletal muscle and the fat cells don't have access to glucose. That helps prevent a further fall in glucose levels. But now look what happens in the liver. In the absence of insulin, the liver starts making glucose. Okay? It breaks down its glycogen stores and it starts synthesizing new glucose molecules, and that's what the actual lecture will be about. All you have to recognize here is that the liver cells will start producing glucose, and so glucose levels inside of the liver cell will become high. And this is the coolest thing, I think. Glucose will move from high concentration in the liver cell down a concentration gradient into the plasma, where glucose levels are now falling and are low, and will replenish glucose levels in the bloodstream so that the brain will have glucose to eat and oxidize. and that these glucose transporters will move glucose independent of, it doesn't matter what direction. All that matters for the movement of glucose through these transporters is that it moves down a concentration gradient. So in the fed state, these glucose transporters move glucose from the outside in. In the fasted state, shown here, glucose will move from high concentration inside of the cell down a concentration gradient into the plasma, where glucose levels are low. This ensures that the brain, again, has access to glucose to maintain its normal function. So the take-home message, in the liver, glucose transporters are always present in the plasma membrane. Remember, this is not dependent on insulin. And so in the post-absorptive or the fasted state, the liver produces glucose, and glucose moves down its concentration gradient out of the cell and into the plasma through the same GLUT2 transporters. And I just think that is amazing physiology. Okay, so this is going to be important. I will recap this again in class with you guys quickly, but since you guys have watched it here, I will assume that you have all been exposed to that material at least once. All right, thanks for listening. Have a great day.